Hello students, today we are going to discuss about the vacuoles which are seen encased in the plant cells. So, vacuoles, as name indicates, they will be in a vesicular form of structure. They are single membrane bound organelle. The membrane of these vacuoles are called as tonoplast. These vacuoles occupy very large space, so these are very large organelle, fluid filled vesicle, which is present in the plant cells. It has been observed that different types of and different numbers of vacuoles are present in the plant cells. They are also involved in or they act as a storage house of the cells in the form of granules. These vacuoles also contain many digestive enzymes. So in that case, we can say that the vacuoles are the lysosomes of plant cell. If we talk about their size, on an average, they are able to occupy 30% of the cell volume, but and on average it occupies 30% of the cell volume, but they may vary from 5 to 90% of the occupation, or they may vary 5 to 90% in their volume, depends in which condition cell is, or depends on the cell in which like in which stage of the cell, in which cell stage these vacuoles are present in the cells. Earlier, what scientists do, they thought earlier when this vacuole was discovered, they thought that the vacuole is bigger in size. So it's better to say in case of plant cell, they are having three main components. First will be the nucleus, second will be the vacuole, and third will be the cytoplasm. But later they said it will be the one of the membrane bound organelle which is present in the plant cells. Like I said, they may vary in their cell volume from 5 to 10%. In immature cells, when cells are immature, means they are in the growing stage of their life, they are having small vacuoles. Or when cells are actively involved in the cell division, they are continuously making proteins, continuously utilizing them for the cell division, next cell division. So in that case, there will be no storage granules, or we can say there will be no storage. So in that case, vacuoles are small in size there. How vacuoles are formed? Vacuoles are directly from the Golgi bodies. They they come from the Golgi body in the form of vesicle. Then different small vesicles join together. Then they form a big size vesicle that is called as vacuoles. As I said, they contain many hydrolytic enzymes. It has been observed that the vacuoles are structurally and functionally related to the lysosomes which are seen in the animal cells due to the presence of their wide range of hydrolytic enzymes. These vacuoles may contain different types, so they are having many transmembrane proteins which are involved in the export and import of the things from cytosol to vacuoles and vacuoles to cytosol. They contain different types of sugars, salts, acid, bases, nitrogenous compounds, and even it has been observed that in vacuoles we are having some secondary metabolites present in them. Now, what is secondary metabolite? When plants are in growing stage, they form proteins or nucleic acids, whatever is utilized by them. These products, whatever is utilized for them for the growth and development, are called as primary metabolites. इन शॉर्ट हम लोग बोल सकते हैं प्राइमरी मेटाबोलाइट्स वो कंपोनेंट्स है जो सेल अपनी ग्रोथ के लिए यूज़ करता है. देन while processing, this is a metabolism process, while in their metabolic processes they secrete some components which are not utilized by these plant cells. So this secretion which are not utilized by the plant cells for their growth and development are called as secondary metabolites. But these secondary metabolites are beneficial to us. It's very important economically, industrially point of view, or pharmaceutical point of view. These secondary metabolites are very important to us. And one of the examples is that these vacuoles contain alkaloids. These alkaloids are one of the example of secondary metabolites. And these vacuoles also contain anthocyanin pigments. They also contain anthocyanin pigments. So these, like, if the pH of the vacuoles can vary, it can be as high as from 9 to 10 pH due to the presence of large variety of alkaline substances in the vacuoles. Due to the metabolic process, there will be change in the pH. Sometimes this vacuole will be having high pH within the range of 9 to 10 due to the presence of large quantities of alkaline substances, or there may be shift pH to low as at the 3 pH. It has been observed due to the accumulation of different acids in different quantities. We can say these may be the tartaric acid, oxalic acid. Okay, so we will see how acids are present in the what are the different, or how acid will be stored in the vacuoles while photosynthesis. We will study this in the functional part. Melite. So this is here we are having typical plant structures, typical plant cells, where we can visualize this, that we are having chloroplast, organelles, plasma membrane, cell membrane, nuclear envelope. Nucleus is at the one side, but if looking at this diagram, we are having large vacuoles. The surface of these vacuoles are called as tonoplast. On this tonoplast, we are having different membrane bound proteins, which are, or we can say these permeases, which allow different types of molecules coming from cytoplasm into the vacuole. So in short, we can say these vacuoles are having different composition than that of the cytoplasm. As we said, they are having many hydrolytic enzymes than present in the vacuoles, so they are involved in the autolysis of many cell organelles present there. So this vacuole will be obviously having tough structure, tough plasma membrane than the membranes of other organelles present in the plant cells. So next part. So Melite was a scientist. He was working on the plant cells in 1975. He observed and he divided vacuoles mainly into three categories. First, he said there are vacuoles which will be called as vacuoles only because they contain simple acid hydrolase enzymes. Or the second, they found vacuole-like structure. They called those vacuoles as Spherosomes, and the third part is the aileron grain. So, as per the malite, we are having three different types of vacuoles present in the plant cells. Now, if we will talk about the vacuoles, we can simply say these are enlarged structures formed by the fusion of small vacuoles or small vesicles which are coming or formed from the Golgi bodies. As we have seen when we have studied how proteins are transferred when they are getting translated on the ribosomes of the endoplasmic reticulum, then these proteins are taken into the lumen of the endoplasmic reticulum. Then from the lumen they are getting glycylation processes happening there. Then from lumen of the endoplasmic reticulum, these proteins in the form of vesicles are transferred to the Golgi bodies. Then again some modifications are there. Then from the Golgi bodies they form simply the small vacuolar or vesicular form of structure. These are called as provacuoles. These are called as provacuole. When different provacuoles join together, they form a large vacuole. They are having different types of acid hydrolases present in them. And while observing or studying these vacuoles under the microscope, many times it has observed that these vacuoles inside these vacuoles we are having different cell organelles. Like it has been observed that there is a presence of mitochondria or plasters present in the vacuole, which suggests that these vacuoles are involved in the autophagy of the plant cells. So they act as a recycle process for us. So next is our spherosomes. So they are round shaped spherical particles, membrane bound. Their size may vary from 0.5 to 2.5 micrometer in diameter. These spherosomes, as we have studied this in the gloxosomes, that these spherosomes are rich in, or they are having higher concentration of lipids and proteins in them. These spherosomes directly originate from the endoplasmic reticulum. When there is the accumulation of oils at the end of the strand of ER, then these like oil particles pinches off from the endoplasmic reticulum in the form of vesicles, and these that time are called as prospherosomes. When they pinch off from the endoplasmic reticulum along with oil accumulation in them, that time they are called as prospherosome. So again these prospherosomes are grow in size, then they form the spherosomes. It has been observed that these spherosomes are involved in the lipid synthesis and storage process. These are involved in the lipid synthesis and the storage process. Spherosome has been observed in maize root tips, or we can say spherosomes of tobacco endosperm plants contain different hydrolytic enzymes. That is why we consider these vacuoles as a lysozymes. And there is accumulation and mobilization of reserve lipids. जब प्लांट्स को एनर्जी की ज़रूरत होती है वहाँ से लिपिड्स बाहर आते हैं, वह जब प्लांट के पास एक्सेस लिपिड्स होते हैं तो इसमें स्टोर हो जाता है, मीन्स एक्यूमुलेट भी हो रहा है और बाहर भी दे रहा है. Accumulation and mobilization of reserve lipids. And also they are involved in the digestion of cytoplasmic components by the phagocytosis process. Now the third is our aileron grain. Aileron grain means this, as name indicates, this is the vacuoles which are observed, which are seen in the grains. We can call these as membrane bound storage particle, or we call these as protein bodies. These protein bodies are mostly seen in the endosperm of the seed and the cotyledons of the seed. As we know, the endosperm is the storage house. When this endosperm is utilized by the cotyledons of seed, then only seed germination process takes place. This aileron layer or aileron is formed in the लेटर स्टेज ऑफ सीड राइपनिंग. जब सीड को राइपन होता होता है, ये तब पहले फॉर्म नहीं होता है, जब मतलब मैंने मच्यूरेशन ना सन सीड्स गसन वाजन तम सच ये लेयर फॉर्म गसन एज यू कैन सी इन द डायग्राम. But first thing, they get disappeared. This layer, aileron layer, get disappeared in the early stage of germination. जर्मिनेशन होगी जब एनर्जी जो है, स्टार्च है जो ग्रेन में, जो फूड वैक्यूल है, वो डिज़ोल्व होगा, तब जाके जर्मिनेशन होगी. सो दैट इज़ वाई दे हैव ऑब्जर्व दैट देयर इज़ अ डिसअपियरेंस ऑफ दिस aileron लेयर इन द अर्ली स्टेज ऑफ germination. So we are having here maize wheat grain, sorry. So this wheat grain is also having aileron layer. If we will talk about the corn grain, they are also having the aileron layer. So this aileron layer is formed when there is the ripening of the seed. In the later stage of ripening of the seed, these are formed, and in the early stage of germination, these layers get dissolved so that there will be transportation of energy from these endosperm to the growing cotyledons. Now, the functions of the vacuoles. So we know that these vacuoles are involved in the storage, as we discuss right now. Even it has been observed that these storage organelles act as both. They can store nutrients as well as waste product in them, and they are also involved in the turgor pressure of the cells. That is what we can say. They vacuoles maintain the rigidity of the leaves. Turgor pressure barabar vacuole that is board bar vacuole, that gives the turgidity to the cell and younger parts of the plant. If we will talk about the storage, these vacuoles also contain potentially harmful products in them. Even there is something specialized in them, that sometimes these vacuoles contain some different products. Like we can say, in rubber is formed in the vacuoles. This is from Hevia brasilensis. इससे क्या होता है, जो इनका जो वैक्यूल है, these vacuoles are able to form the rubber. और another opium के अफीम, and this opium is produced by or stored in the vacuoles, and the species involved is the Papaver somniferum. So these are actually the waste products, but for us these are important products, but for plant these are actually the waste products which are stored in the vacuoles of the plant cells. Even if we will talk about the turgor pressure, these vacuoles are having higher amount of sodium ions present in it, and these turgor pressure was studied in the alga Nutella, where it has been observed that the vacuole of this alga is having sodium ion channels present on the surface of the tonoplast. As these vacuoles contain higher concentration of sodium than the cytosol, as you have studied how opening and closing of stomata is happening in the cell. So even we can say that these vacuoles maintain homeostasis of the cell. Means if there is a change or there is a variation in the environment, these vacuoles doesn't allow plant cells to get affected by the change in their environment. If we will take one example, when there is pH drop in the environment, or we can say when there is high pH in the environment, what these vacuoles do, they accumulate much of the H positive ions in it, so that we can say that these vacuoles act as a buffering system इन द साइटोप्लाज्म, क्योंकि जब ज़्यादा हाई पीएच होगा तो ज़्यादा एसिडिक कंडीशन होगी, ये वैक्यूल्स क्या करता है, ये ज़्यादा से ज़्यादा एक्यूमुलेट करता है H positive ions. Even we can say that they also are involved in the breakdown, like we will see this later. They are involved in the breakdown. जैसे जो ये है लाइसोसोम्स, ये क्या करते हैं, किस में ब्रेक डाउन करते हैं, लाइक आई सेड ऑटोफेजी, जो ऑर्गेनेली को मतलब ब्रेक डाउन होना या उस ऑर्गेनेली में कुछ प्रॉब्लम है या उसकी एज हो चुकी है, वो क्या आता है, वो लाइसोसोम्स के पास आता है, लाइसोसोम्स उसको कवर करता है, उसको एनगल्फ करता है, फिर उसकी डाइजेशन करता है. सिंपली दिस इज अ ब्रेक डाउन. सो दीज लाइसोसोम्स आर इन्वॉल्व इन द ब्रेक डाउन एंड रिसिंथेसिस ऑफ पॉलीमर्स. वो रिसिंथेसिस करता है पॉलीमर्स की, ब्रेक डाउन हुआ फिर रिसिंथेसिस करता है. इवन ये जो है फॉस्फेट प्रेजेंट इन द सेल्स, these phosphate are stored in the vacuoles in the form of polyphosphate. इवन उसकी वजह से क्या होता है, जो हमारा ये टर्गर प्रेशर मेंटेन करता है, उसमें डिफरेंट, इवन इट हैज़ बीन ऑब्जर्व दैट द tonoplast layer or the मतलब plasma membrane or the vacuoles are having different transmembrane proteins which are involved in distinct functioning, like there are specific sugars or amino acids and other metabolites which are able to cross the lipid bilayer of the vacuoles. Even we can say जो quality है or the quantity of these vacuoles differ from that of the cytosol. So if when this is larger, as we have seen in the diagram, this vacuole is larger in size, this due to its size they are able to give mechanical strength to the cell. Even one important function, as I said earlier, that they are able to store some acids. So in succulent plants, what are succulent plants? These are those plants which don't open their stomata during the day. What is happening, because there is a less availability of water, what these stomata do, they open at the night and take the carbon dioxide in. When these stomata take the carbon dioxide in, then the plant cells convert this carbon dioxide to the malic acid. कार्बन डाइऑक्साइड अंदर लिया किसने, स्टोमेटा सेल्स ने, इन्होंने इसको कन्वर्ट किया मैलिक एसिड में. अब ये एसिड है, what this will store, this can't be stored in the cytoplasm or in the cell, then this malic acid is stored in the vacuoles. When on the next day, when light is available, light energy is available, then this acid is converted into the sugar. Even that time stomata don't get open up, they remain shut, they remain closed that time. Even these, in case of storage function of the vacuoles, proteins are stored in the vacuoles for, it can be stored for years, as we can see this in the seeds. So these were the structure, how the vacuoles are having different structure, what are the different types of vacuoles, and the functioning of the vacuoles in the plant cell. Thank you.